Okay, can you see my screen? All right, I think we should get started. So good morning, everyone. Good afternoon, wherever you are. As usual, this meeting will be recorded. So if anything occur during the meeting, please let us know, and we can make edits before we post it on YouTube for sharing across the world to people that can't attend due to time zone differences. So the agenda for today is we'll start with a reminder of our plans for data collection at the Allen Institute for this community project. I present a few slides, but we have seen before just to bring back context. And then Severin will speak about our team's progress in collecting the pilot data that we agreed to do a few months ago. And then I have a really brief update on where we are with the data collection SLAP2 platform. And then I'm hoping we can finish the meeting with a discussion of analysis of the NeuroPixel data that is now public to help us move towards the data productions in early January. As usual, you can see the agenda on our website, and if you click on the link at the bottom, it brings you to a discussion page where you can discuss the meeting and see the content. And so I posted yesterday the slides that I'm about to present. Okay, so let's start. Slideshow. Can you see the full screen? Okay, so as a reminder, our goal through what we identify in the review is to cover different set of mechanisms, and so what in the experiments that we design, we aim to create a different number of prediction contexts, and which are listed in the review. And this is just a reminder from a figure from the review. And the design that we converged to had 4 types of session. A standard mismatch, where a simple drifting grading is presented multiple times repeatedly. A sensory motor mismatch where the mouse is controlling the drifting grading of the drifting grading speed by walking forward. A sequence mismatch where sequences of orientations, drifting grading orientations are presented in a specific order, and we alter that sequence. And a duration mismatch where the duration of the drifting grading or delay is varied. And so the pilot that we did with the NeuroPixel team and some of the SLAP2 data was to aim at testing all of those sessions and help answer a certain number of preliminary questions. Our current goal is to have this essentially five cohorts, 2 for 2 NeuroPixels, 2 for NeuroPixels, and 1 on SLAP. SLAP2, I apologize for the typo here. And we are aiming to have, for each 2-photon rig, we'll have 2 cohorts, one where the mouse is trained on the sensory motor context and then see all of the 4 types on the rig, and another where it's trained on the sequential context and then again see in the different order the same context. And the same design on the NeuroPixel. The difference here on NeuroPixel, we only do 4 recording sessions chronically. So that's the intent. And on SLAP2 platform, we started collecting the same design, focusing on the sensory motor exclusively. So that's our current walking model. And a few months ago, we shared with all of you our plan for the pilot analysis. And as you can see, we were planning to collect some of the first earlier data in October and November. And so our goal is to have insight to make any modification that will be needed for the productions in early January. And we are still working on that schedule, and we work very hard. I'm very proud of the team, extreme effort to be on that timeline. And so today we have data set to share with you. And so, NeuroPixel, maybe Severin, do you want to speak to that?
Yeah, of course. Let me share my screen.
Before Severin, any questions on this context in the group?
Jerome, quick question. So
Yeah.
all of those are recorded now with the sensory motor. So all of those are.
That's right. So
yeah.
that's what Severin is going to share. We in the
Yeah,
pilot.
I'm going to speak about that now.
Okay.
Yeah,
Thank you, Severin.
of course, of course. So yeah, for those who hasn't seen me yet, I'm supervising the data collection with the NeuroPixel probes. I wanted to start, as Jérôme just mentioned, to say a big kudos for the two RAs that work really hard on this collection of data, which are Ryan Gillis and Casey Lennon-Jones. So yeah, really big kudos to them. It was not without challenges. So yeah. So basically what we did two pilots. They are both SST-AI 32. We did 4 days experiment with each of them, and we had 6 probes inserted, except for the last 2 experiment where we were missing 1 probe. Um, Basically, the next step here in blue. So we had to delay a little bit the data collection of 1 week because we had some probe broken. We had to replace like all the probes. So yeah, so that's a little bit further in time than the previous slide that Jérôme showed. But then after the experiments, there is the perfusion, which is generally the day off or the day after. There is a clearing method, which is live canvas that lasts 3 weeks, so it's quite long. The imaging can happen anytime depending on their workload, basically. Once the data is uploaded, the spike sorting happen in 1 or 2 days. The data conversion can happen in few hours, and then IBR app, which is the application we use for making the boundaries of the areas. So that is done in few hours for each brain. Just to summarize also, for these 2 pilots, we did only the cohort 1. So they were trained on the sensory motor context, and then they had sensory motor, standard oddball, sequential context, and temporal jitter in this order. Okay, so I wanted to start by reminding you what the implant looks like and what it looks like on the brain. So the first picture shows you the implant without the probe. The second one with the probe inserted. So the black thing that you see here are the probe that are very near to each other. So it's really a dance of probe, basically, to try to have the probe at the right place without colliding together. And then Ryan really worked hard on this. And basically, this schematic here shows you which area we aim to target with each hole. So we have 2 holes with ACA, and this hole correspond more to the what is known in the literature. So, and this one, we have used it extensively, so we know we get ACA and MOS and other areas. This hole here is for VIS-A. I will show you a little bit later what it looks like. This is for VIS-P, VIS-LM, and then this hole here is for MOP. So in the best of the world, all of these implants are perfectly matching the maps and perfectly matching the CCF, but it's not always the case, right? So, but now I'm going to show you 2 cases for the 2 pilot where the implant worked out really well. So here in gray, you have it's the eyesight maps over the brain of the 2 pilot that I just showed you. These are the numbers of these 2 pilot, by the way. So the goal was to have a hole near the center of retinotopy of VSP, which is the case here and here. The center of retinotopy is like this yellow and blue lines here. We wanted a hole that is also near the center of retinotopy from LM, which is here and here. That worked out really well. And then finally for VIS-A, so A doesn't appear in the eyesight map, but we know where it is. So basically, we did a study before where we had to record from Vis A, so we have some experience with it. So basically, we know where is Vis AM, and we know Vis A is outside of Vis AM, between Vis AM and RL. And most of the time, Vis A actually appears like in RL, in the most anterior part of RL, in the ISI map. So that is an example of a targeting that we did. So these are actually, the little crosses are actually where the probes went. So you don't see with the SI map, you don't see the frontal parts. You just see the visual cortex part of the brain. Okay, so the session. So exceptionally, because we had this breakage of probes, we had to push everything of 1 week. So both mice were habituated to the rig, meaning they had a full session on the rig every day for 2 weeks, so that they get used to this environment. And we also make them wait a little bit, so they are used to waiting a little bit before we start the experiment, while we do the insertion and the stabilization of the probes. So we had 6 sessions out of 8 that passed the QC, and the 2 that didn't pass were the second mouse, were the standard oddball and the temporal jitter. But all of the other ones now, as Jérôme mentioned, are public and they are uploaded. I just wanted to tell you that these recordings are very stable. So these are examples of firing rate for 4 different probes in one of the days of recording. So you can see, and here is plotted the spike count per second as a function of time. So you can see that the traces, the spike count is very stable over time here. And it's also stable in terms of drift, where the ideal experiment for us has a drift that is lower than 50 microns. And here again, example of few probes during one of the recording, when you can see here the drift that are plotted with the number of units there. So you can see that they are all like below 50 microns, which makes us very confident that these recordings are pretty stable. Now we had I don't think we managed to target LGN with the VIS-A probe, which is surprising to me, but okay, here are the graph of these are the channels here on the x-axis. On the y-axis is the unit count. So in green you have the good units, and in bad the noise. So these are for 4 different probes on 4 different days. No, yes, no, 2 different probes on 4 different days. And so here, this is the hole in the VIS-A hole. So if we got LGN, we would have some population of units here at the tip of the probe, which we don't see here and here and here. Maybe here a little bit. So I was kind of puzzled by that. But if I looked at the VIS-P hole, I get this population there at the tip of the probe. And it's either, in my experience, it's either LP or LGN, and we don't have the receptor field yet, so I cannot tell you if the receptor fields are small or big for this population of cell at the tip of the probe. But basically, I looked into pinpoint, and when you're very anterior in the hole of V1, you can reach to LGD. So it's not impossible, but we will confirm that with the receptor field and the anatomy afterwards. Yeah.
You have a question from Kareem in the chat.
Of course, yeah.
How do you estimate? Yeah. How do you estimate the drift distance?
I think it's all in the spike sorting. So I don't know the detail of the spike sorting algorithm and everything, but it comes out of.
I think you can measure how the probe
Yeah.
moves on the shaft, right?
Yeah, you see, yeah, you see the shift. I don't know. Honestly, I don't know exactly the algorithm for that. I'm sorry.
But it's one of the metrics that is created by
Yeah,
Kilosort and is available
yeah,
for everyone.
yeah,
Okay.
yeah, it is.
Gotcha.
Yeah, yeah. It's in
All
the
right.
QC. It's in the sorting QC. Yeah.
Thank you.
Sorry, I'm not specialist in
It's okay.
spice
I was just curious
sorting.
because I know we typically do the 1/r roll off of the amplitude, so I thought you might have something different that, but yeah,
I
thank
see.
you.
What is your usual kind of drift that you see usually with your recordings?
Typically around that, yeah. I mean, it's depending on the length of the probe. The longer the probe, the more stable it is usually. Shallow
Yeah.
probes typically can drift a little bit with brain movement.
Yeah.
But we typically have this 1/r amplitude decline. The further you are from the units, the more and you can correlate that spatially across different adjacent channels.
I see. Yeah.
Thanks.
Note that in this instance, we are actually going to share the raw data. It's already public. So Carter will speak to that after 7.
Okay, great. Thank you.
Okay, so another topic I wanted to touch is the running. So the two pilots didn't run that much. They basically walked a little bit uncomfortably, and they moved, but not as much as we would have liked them to move. And we talked about that, and apparently there was a decision made that we should not have a wheel in the cage of the mice. But all of us discussed this, and we think that it would be better actually to have a wheel in the cage before the experiments, so that they just are used to learn on the to walk on a run on the wheel. And the more they will walk or run on that wheel, the more they will run during the experiment, with the caveat that sometimes some mice don't run; they just don't. So I would like to be sure that everybody is okay with us like putting the wheels in the cage before experiments. So let us know if you have any concern about that.
I think it was
Uptotype,
mostly
yes,
concern from Sarah. So if Sarah is not able to speak, oh yeah, yeah. Sarah said the concern is that the animals are trained to a visuomotor mismatch in a sense. Yes, that's fair. I think our experience, Sarah, is that adding the wheel to the cage makes actually them walk and run on the disc because they're habituated to do that. And so it's a trade-off.
Yeah. Like already the mice that are passive usually have more trouble to run than the active mice. So if they're on top of that, don't have a wheel in the cage, they really will be very uncomfortable during the experiments. So think about it and just let us know what you think.
Question. So is this primarily to make them run more during the closed loop sensory motor session?
Yeah.
That would be the intent. Yes.
Yeah.
But then, right, and all of the sessions with mouse will be on the disk.
Yes, they are all on the disk. Yes.
Okay.
They are free to run, not run, walk, not walk. They're free. Yeah.
Okay.
Note that we have the wheel data, and so you all will have access to the wheel data in the NWB files, and so I think it will be interesting to see because I think the mice they still engage with the wheel, but in a much less running way, so that they can still move it slowly, and so I'll be curious to see if regardless there are responses to the oddball. In this context, yeah. So this is something we could analyze a little bit before making a decision.
Yeah.
Can the wheel speed be constrained in cage and free in rig? You mean like limit put friction? in the wheel?
Yeah, exactly. So to deal with something that could alleviate Sarah's concern is that you make the wheel in the cage cannot go beyond a certain speed, so they can only walk on it. So induce more friction, but then to induce the visual motor condition in the rig, it has less friction, so they can eventually run.
I don't know if there's a device to do that. Do you know of a way or?
I don't know of a way, but it's not hard to do. Basically, it's the rotation of the wheel that you can put some
But
brakes
you-
on it.
Would you create an electronic device to do that, or
No, it doesn't have to be. It's just that the wheel on the cage might be equipped with some, uh, you know, a disc that has some friction, for example, so that it kind of offers more resistance to the animal if they jump on it and run. So they can't run really per se, but they can walk. The idea is to strike a good balance between the, no running at all versus running at full speed in the rig. So something in between in the cage.
I don't see how that would do away with the prediction error, though.
It wouldn't 100%, but the idea is to try to habituate the animal so that all of them would end up running at some point or locomoting in the rig.
I think it's a good point, but I think I think we're not giving mice enough credit here. I think that mammals have context specific learning, and it happens immediately, and it's not like, so like, for instance, all the hundreds of papers that have mice lick left or lick right for a reward, it's not like when their mice are in their cages you keep them from licking because you don't want them to accidentally learn that licking doesn't lead to a reward, right? They learn when they're on the wheel that a lick means a reward. They can separate context here. So I would not be terribly worried that having a wheel in their cage is going to keep them from learning the context specific visual motor coupling. It's not like they have a big monitor in their cage that's blasting stimuli in their eye like they do when they're on the rig. Anyway, like, I think it's a pretty smart point for sure. But then I also would not be terribly worried that this is going to keep the mice from learning sensory motor pairing in the specific context.
No, I'm not saying they're not going to learn or they can learn faster. I'm just saying that within the context, just to shorten the habituation, if that's a concern across, because some mice don't run at all, and most of them learn the condition. So if we standardize this across all animals, might be a way to shorten the time a little bit. But again, we're not doing the experiment, so I let the people who are actually doing the experiment speak about, the however many animals or the percentage of animals that they see needing to.
I don't think, Karim, I'm not disagreeing with you. I'm addressing the point of whether or not to put a wheel in the cage or not.
Right. Yeah, I get it. Yeah, I understand.
Yeah. And I just don't think, I think it's a I think what they're doing, I think just having it in there, letting them freely run on it should be totally fine. But I don't know. I mean, they're getting visual prediction errors all the time, in their experience. Like suddenly a mouse comes up behind them, like that's a visual prediction error. Like it's context specific, and I think that's part of the circuitry we're studying.
Well, Jordan, thank you for so much, and Karim, thank you for your perspective. I wonder if Sarah's concern about based on any data analysis that was done or like something that is established in the literature. I think because this is one way to go at it is like check if there are data that will support one way or the other, and the second is any control experiment that we might need to do to alleviate this concern. I feel that, yeah, there's several ways, but I wonder if, Sarah, if you have any data on this question. And if it's too difficult for you to speak, maybe let us know and we can continue the conversation on the forum. Oh, okay. So I given that Sarah is a little hard to connect. She was in the tube. I think we just should move this conversation on the forum, and then have an in-depth discussion there, so that I don't want to sort of surface discuss it. I think Jordan's point is important, and I also want to respect Sarah's perspective. So let's just move.
If I can just say something, it's gonna be super hard to get wheels that make them walk but not run. We don't have that right now, and I don't know how to make that happen at a large scale also. But yeah, please continue this discussion in the forum and let us know what you want us to do. Okay, I will continue. So now there's one bad news, basically, is that the opto-tagging didn't work really well. So here you have like the pulses of opto-tagging, so a long pulse, shorter and even shorter pulses. And then you would expect to see responses here under the pulses of lighter color. As you can see here, there's barely any response for these 4 probes here. And that was the case for each experiment for the 2 pilots. So what we did, we were a bit puzzled because, like, I can show you. So this is a test that we did with another mouse the week before, where we actually had opto-tagging response. But then when we did our experiment with this, the pilot, then we didn't. So what we did is first we changed the opto-tagging, optogenetic fibers. We had even more power, a little bit more power, which was like welcome. And then we also were wondering about the genotype of the mice, because they were brothers and sisters, and we even recorded from another sibling so 3 siblings of the same litter, and all of them didn't show any opto-tagging response. So we were a bit concerned about maybe the genotype. So we did an ear clip of the mouse before the perfusion, and and then the team that takes care of the mice looked at the genotype and confirmed that the genotype was actually okay. But we are concerned about the expression of in these mice, yes. So what we're going to do is we have been given 2 SST AI32 mice from a different litter, and within the next few weeks, we're going to record from them and see if we get like a response from opto-tagging, or if there's... So also Brian did another test where he... So we were thinking maybe the 6 probe make a shadow and prevent the responses, but then he made the test where he removed all the probes and he kept 1 probe, sorry, and then removed some of them, and then did the opto-tagging and obtained nothing again. So it's either these mice have a problem of some sort, but we're going to figure that out by testing other SST mice in the next few weeks, as I said. So what we learned from the pilots, I think it's very important, is the debate about the wheel in the cages. We have to do more tests for the opto-tagging, which unfortunately didn't work. And also what's happening with the LGN on probe C and see the receptive field. So Carter will probably speak about that a little bit. That's all. And also we filled up some notes, so you will have also for each experiment you have experiment notes that details exactly what we observed during the experiments. So yeah, that could be very helpful also. So that's all I have. Any
Thank you, sir.
question? I don't see. Yeah. Now yeah. No question.
It was a wonderful presentation, sir.
Oh, cool. Thanks. We're very excited. It's a great project. We're very excited to get some good data from it. Yeah.
Okay. Any question? Okay, so Carter is going to introduce the how to access the data and all the packaging that he did for you guys. Carter, maybe introduce yourself and then take the screen.
All right, let's see. Hey, everybody, can you guys see this?
Yes.
Okay, cool. Yeah, I'm Carter. I don't know if I've already introduced myself in a previous meeting, but yeah, I'm an analyst on the OpenScope team. So I've mostly been in charge of packaging and processing our data. So I'll just show you what we have. We have 6 sessions, and everything you need to access them should be accessible on the website here. So I'll just show you. If you go to the website, first of all, you can see the experiments tab where we found the SLAP2 files. You can see, all right, we have these 6 EFIS files here. And then if you click on them, Ryan and Casey were great and wrote all these detailed notes. So you can see the experimental notes that they took when they actually ran the session. And then we've added this section here that shows you how to access our data. So just like our SLAP2 files, our data is accessible on Dandy. It's a different Dandy set because the data is a different modality. But you can find these here. You can also find them on Dandy if you just look up predictive processing. You'll be able to see it. But yeah, so we have the 6 files on Dandy here. And then also, they're in NWB format, just like the previous ones. But because they're a different data type, the NWBs are structured pretty differently. And you can, if you're unfamiliar, then we wrote up this little notebook here that will just give you guys an introduction to accessing the data. So you can click that link on the experiment page and it will just go over the main components of the NWB and just gives you the quick code to sort of access the necessary fields. It also has this cell that will allow you to download the file directly from Dandy if you want to do it programmatically. So the main pieces of the file that we have are, oh, skip that. Let's see. I embedded the entire file here. One sec. Here, if we go to analysis, you can see the notebook will also display on the website here. So this is probably a better view. So it shows the whole NWB file here. We have the subject metadata. We have the stimulus information, which are in the form of basically a set of tables. These ones here are the opto tagging, the pulse trains, and the raised cosine presentations. We have our new receptive field stimulus and our traditional receptive field mapping, and then we have control blocks and then our sequence mismatch. So you can access those easily. You can see the table and has all these columns and rows. Probably the most important one you want is the trial type column here, which will tell you whether or not it's a mismatch. So the values for the trial type are as follows. We have the LFP. So this just pulls out the LFP along the probes. And then other than the stimulus, the most important thing here is the units table. So this is, each row is a unit from kilosort 4. And then the columns are all these different metrics recorded for each unit that was pulled out. We have the spike times. Yeah, then you could use these for filtering units however you want. And I believe you guys asked about drift. I think that's going to be encoded somewhere here too. Yeah, there's the standard deviation of the drift And there should be other ones here too. Yeah, so please look at that. For instance, you can just pull this out and then get the distribution of the signal to noise ratio of units here, and then you could select them accordingly, etc. You could pull a unit out and you can get the spike times. So this section is not going to be too useful now because we don't have our CCF the coordinate space information packaged into the files, but we will in future versions. And then this code will be very useful because getting the CCF location for a given unit is not intuitive in NWB. So you're going to need to use this code here. And then finally, we have eye tracking. So you can see the trace of the eye through time. These are relative positions, not absolute positions. And then we also have the running speed.
Carter, Sarah
Yeah.
has a question in the chat.
Oh, yeah, let's see. Yes, we do have waveforms. So I'll actually go back up here. So they have a waveform mean for each electrode that unit was detected on. So this is actually an array of waveforms, and you can get the max waveform. Well, the code I just showed you here, this will actually get you the waveform means here, and then you can get the waveform with the minimum value. So presumably the peak waveform with this little snippet right here. So yes, you can get the waveforms for every unit. And I think Kilosort actually uses the waveforms as a way to determine whether or not the unit is high quality or not. So that's encoded in here too somewhere. Yeah, I would look more into the details of Kilosort 4 if you're interested. And then, and then after the eye tracking, the last thing is the running speed. So I encourage you guys to look at these. Maybe we can do some data-driven analysis to determine, whether or not these mice are running enough. So that's this notebook here. And then the final thing I wanted to show you guys is our raw data and our CodeOcean pipeline. So if we go to the experiment here, we have an additional link. This is not present for our SLAP2 files. It's linked to this CodeOcean public collection. So we were able to get this data up in about 2 to 3 weeks after running the session, which is record time for OpenScope. So we're pretty happy with that. And I think it'll be faster going forward. So we have this little collection here on CodeOcean. I'm logged in, but you don't need to be logged in to access this because it's a public collection specifically. So you should be able to click the link and then you'll be able to see this little set here of the data. And these are unpackaged and partly unprocessed. So they're going to be pretty different from what you see in the NWB, but it does have the output, the direct output from Kilosort, and then it has basically the raw movies we recorded, and then basically output from our experimental rigs. If you're more interested in exploring this unprocessed data here, then I encourage you to talk to us about it. There's also these useful metadata files, which might have more bespoke information if you're interested.
These include behavior videos of animals.
Yeah. This should have the videos in here. So yeah, actually has the eye video, the face video, things like that. And I guess finally, I should just show you the packaging pipeline that we actually use here. So this is what we set up on CodeOcean. In the future, I think Val and we have plans to make our processes more transparent. So in the future, we might release these, and you can actually see the code that we use to do our packaging and our post-processing stuff. But yeah, are there any other questions?
Question. Thanks, Carter. That was a very nice intro. So to start, would you suggest we go with the Jupyter notebook that accesses the NWB files? Is that the easiest way to?
Yeah, that will be, you can just plug in the file or the file path if you've already had it downloaded, and then it will basically pull out all of that information for you. And then you should be able to start your analysis pretty easy.
Awesome. And if you have questions, should we reach out to you?
Yep.
Would that be fine? Okay. Thanks.
I want to emphasize the Codison work it was a lot of hard work from Severin's team and Carter collaborating together to push the data out so quickly in the raw form in a cloud system that everybody can see. And it's the first time we are doing this for OpenScope. So giving you that like unrestricted access to all of the raw files. And what we hope to do in the future is use this because CodeOcean comes with a whole analysis ecosystem that you can leverage. And so in the future, we'll be able to share with you capsules that contain pieces of analysis that are running directly from those files, and then you'll be able to replicate the environment and so on and so forth. So if you're interested in exploring, that'll be a great feedback for us to see how it works and what you like about it and how we could improve it, because we anticipate to use it very much. It's a whole analysis community sandbox.
Yeah, it turns out to be a very powerful tool for getting our data through really quickly. So
Okay. Any question before we move on to the last few slides? I want to keep time for these. Okay. Okay, so now we have data. I wanted to highlight a few things. So you might recollect we had a number of discussion about Zebra, TreeP, and receptor field. And so those files, this is an example of the session we presented, contains an example of 5 minutes of Zebra, an example of 5 minutes of TreeP, and then 5 minutes of a couple of patches. So I'll personally will be really curious to see any analysis of comparative analysis of those stimuli, particularly about receptor field mapping, because ideally we should be able to remove the couple of patches if Zebra is fulfilling its purpose, and that might free us some time for the production to, for instance, run Monet, which give us global motion. So comparing global and local motions in the visual cortex will be very interesting, I think. And so I'll be curious to see if someone wants to take this on. That's the first thing. Before we move to the analysis session, just want to quickly update. This is going to be very, very brief on SLAP2. So Andrew Shelton couldn't make it today, but he is with us in spirit. He collected the majority of the data that we acquired in the last few weeks. And so this is reflected on this sheet here. It's a public sheet that show our schedule for SLAP2. And so you see we plan to collect 10 mice, half with glutamate imaging and half with voltage imaging. And so because we have to plan everything months in advance, we have that pan out until March. And we are Thanksgiving week. And so those experiments have been done. And so that means that we've done 2 mice worth of data with the sensory motor context focus on single cells. There were a number of SLAP2 issue, I think, that delay some of the data collection. So the microscope had some issue a few weeks ago, so we had to push it a little bit. But we were able to, thanks to Andrew's effort, to finish this up. And what else? Oh, one of the session, I don't think we could continuously record the same neurons. We had to switch on to another cells. These data is not yet packaged because we were really focusing hard on the NeuroPixel packaging with Carter. And so that's going to be our next priority is sharing those data assets. But we are sort of midway through this process. One of the mice we plan for recording was aborted earlier. There was some health issue, and so we monitor our mice very carefully, and so we decided to interrupt this experiment. So that means that after Thanksgiving, Made is gonna record the next one, and then our plan is to continue finish the rest of the mice after the Christmas break currently. So that's where we are. But you should expect two more mice worth of data. These are chronic recordings, so we're going to record pyramidal cells across multiple days on the SLAP2 instruments with the full sensory motor session. So it's a longer session. Also with the zebra entropy stimuli, notably. So that's where we are on SLAP2. Any questions on this?
This is really exciting. Jerome, one question. So did I understand correctly that in the NeuroPixels you have all different oddball sessions in there, or is it only the sensory motor?
Right. So that's essentially this. So you see here in the NeuroPixels, we are training the mice on the sensory motor. This is the pilot we did. We did this cohort. So we trained the mice on sensory motor, and then on the first day we do sensory motor, and then we switch context.
Right. Okay.
And SLAP is the same.
Okay. So and for now it's just the sensory motor context that's uploaded.
That's right. That's
Okay.
right. We had to decide. This was a pilot, right? So what we hope to do what we hope to achieve in the next few weeks is engage with you guys on the analysis of this data to learn as much as we can before we do more experiments in January to influence the future.
Right. Yeah, that makes sense.
Okay. And so this is my last slide. Karim?
Yeah, I was just curious to know if the voltage sensor within the same cells that we're recording glutamate in has worked. Because I remember Casper at some point said it's still work in progress.
Yeah, do you want to speak to that, Casper?
We have no plans as part of this project to do dual voltage and glutamate imaging. We are working on that, but we are doing voltage imaging with a green indicator, or a yellow indicator rather, and separately glutamate imaging with a yellow indicator.
Thanks.
I'm sorry for the echo. It's my headphone is open shell, and so it's actually crosstalk even if we have headphones.
Yeah.
Okay. Sarah, do you image the same or different neurons across the mismatch condition on the SLAP2? Okay, good question. So we are trying to image the same neurons, and I was working closely with Andrew on those experiments, and I think one of the mice, it couldn't keep the same cell. And so we're learning. I think it couldn't find the same cell. I think probably suffer something in between session, which we unclear. But I think in one of the two mice, we might have followed it through. I'm gonna send up all the notes about those sessions. I don't think we've updated them yet. But the best case scenario is we image the same cell. But obviously, if you lose the cell, we'd rather record something useful than stop the experiment. Okay. What I'm hoping to achieve, and so what would be great for us is organize a little bit any analysis that could be done on those NeuroPixel data. And I mean, I have some questions, and I'm sure you might have a difference. And so I listed them on the screen. We would like to know if there are any issues with temporal alignment. So with Carter, we actually started to plot receptor fields. So we know we see receptor fields. So that is comforting for us that the temporal alignment is correct. But temporal alignment is a delicate thing that, and we've deployed Bonsai on our rig, so that changed how we do alignment. And so I want to make sure that there are no issues. And so any analysis of functional responses will be very valuable for us because we'll sort of confirm the temporal alignment process is done properly. That's the first bullet point and probably the most important to make sure it happens. The second one is I'll be very curious to see a comparison of TRPE, Zebra, and Gabor for receptive field. I think it's probably going to be easier to do Zebra versus Gabor because there are analysis code to do both available. TRPE, I think, is a more niche stimulus developed by the Micron project, so I don't think they have a down receptive field analysis on it. It's possible that it might be simple, but I'm not sure. And so I would like to know if we can replace Gabor with Zebra entirely. Then obviously some efforts could be made towards analyzing the oddball responses in a different context to make sure that our training process worked. I'm a little concerned about the duration mismatch protocol because I think there were a number of discussions that occur between Sarah, Farzaneh, myself, and others. And I implemented a version of this, and I want to make sure that this is working well. And then scientifically, I'll be curious to see any response in the prefrontal cortex in particular, because for us, it's a less explored area than the visual system. And so if anybody is interested in there, I'd be really curious to see. So that's my last slide. I'm curious if there are folks in the room that are interested in some of those questions. I think we should try to get organized in the next few weeks.
Jérôme, can I say something a second?
Yeah.
So for the receptor field, guys, so you will see receptor field on probe C and D mostly. It's going to be VSP and LM. Vis A might have some receptor fields, but my experience is that they look a little weird. They look a little bit like the RL receptor fields. But mostly if you test like your comparison of TRPE, Zebra, and the Gabor, so concentrate on probe C and D basically.
And Carter has some first notebook to look into this. We did, because for us, receptor field is the first line of defense, and so we look at it closely with Severin. And so we have some start, but we can share it with you. Any questions?
A question related to what Severin said. So the metadata will include information on which probe has which areas? Because I guess when Carter presented, I can't remember if that was
No, it's going to take a little bit of time. That's what I said at the beginning. Like you have 3 weeks of clearing and then some weeks of imaging. And then after you have some processes to do to convert the data to be able to open it with the IBL app. So all this will take more than a month for sure. So for now, we are very sure about VSP and LM because we have the ISI map, right? So you've seen like the insertions are actually really in these areas. So these ones were sure. The other one will have to be determined with the anatomy later on. Yeah.
But Severin, can you clarify how you expect people to know which probe to look at?
Well, the visual probes, right? Like it makes sense. So
but I'm just the metadata contains the number of the probe, right? And they refer to your presentation to see which one is which. Is that right?
yeah, it is. I can go back to it for a sec. So I don't know how this is conveyed on the metadata somehow, but like this whole here is for probe A, this is probe B, probe C, probe D, probe E, and F. So, and these are the area that we're supposed to target with this. So the only one that we're super sure that we are targeting is from probe C here and probe D, where it's LM here and VSP here. And the other one, we don't have the certainty that we managed to target them yet. But I don't know how you find the probe letter with the area. I don't know if this is, where is it, Carter,
Carter?
in your, yeah.
It looks like, so we have it in the session JSONs, but I don't think it's, let me see. It's by, oh yeah, actually, let me share my screen here. So,
Yeah.
so if you go to this link on CodeOcean, where we have the raw data, like I sort of briefly mentioned, we have all these metadata JSON files here. So if you click on session JSON, that includes basically all the metadata from the experimental session itself. And so you can see we have this ephys modules object, and each one has a sort of name. So this would be probe A, and it includes the primary targeted structure. So this is the extent of the information we have in the metadata. So for like probe C, you'd be able to see, or probe B here, you can see the primary targeted structure. But I think this is everything we have.
This was not copied in the NWB, Carter?
No, I don't believe so. It's in the metadata. Yeah.
Should probably try to fix that.
Yeah.
So we need to share that. If I understand well, you need to know which probe letter corresponds to which targeting area, right?
Yeah, that's encoded here.
I don't see it, Carter. Where is the targeting area name?
Oh, yeah, maybe not. Hold on. Just has the atlas name.
Oh.
So it might be good for the time being that we put on the forum, Seb, if you just sort of add a little table to include this information.
Let's give a table that gives the association.
Yeah,
I think that'll be clearer for everyone.
Yeah.
Thank you, Farzaneh, for this question.
I think honestly, to miss ACA, it's going to be difficult. What I'm mostly concerned is MOP, because if the implant is a little bit shifted, we might have MOS instead, or I don't know. And then LGN, I'm not sure if we're going to manage to get it. So this is something we need to look at. Yeah.
Okay, it's 10 o'clock. So I propose I will add those discussion points on analysis to the forum. And then you can all go there and express interest in if you want to analyze any of these questions or add new ones. Because I just want to facilitate people analyzing different aspects and transparency between one another. And so this way we can form a plan together on how to construct feedback on those pilots. Does that sound good? It's important to us that we sort of are data driven, and I want to make sure that we learn as much as we can because these experiments are a lot of effort. So, okay, so I'll head the forum, and then we can continue the conversation over there.
Jerome, just one quick comment. Since there's quite a bit of analysis to be done, and there is a lot of data, why don't we just at least draft some plans so that people complement one another as opposed to repeating the same analysis?
That's right.
I think that's going to be a lot more productive.
Okay, so do you think we can form this in the forum or?
Yeah, it doesn't have to be in a live meeting unless necessary. Maybe every lab would have their own interest in looking at certain aspects of the data. So I think the forum can be a good place to mediate that.
Okay, so let's transfer that because I think analysis takes some time to reflect, and a meeting might not be the right places to form those ideas.
Right.
Okay.
Thanks.
Let's do that. And then based on the discussion, can schedule the next meeting because we're going to continue data collection on SLAP 2, but the next time we meet, we should probably be focusing on the data. Okay. Well, first, thanks all the people that collected this data. It was a lot of work. And amazing that it's getting together. Thank you.
Thanks all. Bye.
Bye.